Yeah, so my name is Noma Adaku. I'm currently a fifth year MD PhD student in what we call the Tri-Institutional MD PhD program. So we're kind of a mix of three different biomedical research institutions on the Upper East Side. So I'm currently working on my PhD at Rockefeller, where I'm studying melanoma metastasis. So I'll talk to you guys today about what is cancer metastasis and how we actually figure it out in the lab. And then after I'm done with my PhD, hopefully next year or so, I'll go back to med school and finish at Weill Cornell, which is across the street, and I'll finish my last 2 years in medical school. And then the goal is to kind of keep finishing medical training. So you still have to do like residency and fellowship and all these other things. And if at the end, when there's time for Q&A, if you guys want to ask me about like the career path, I'll be happy to do that. And then, yeah, the goal is to, exactly become a physician scientist, see patients while at the same time having a lab where I'm actually studying the things that my patients are experiencing. And kind of in terms of like non-career things, so I'm originally from the Bronx. So I don't know if we have any Bronx people here. So originally from there, and then I lived there for a few years in Atlanta before going to undergrad at Yale. And I know you guys attend like an early college, high school. So I actually did a very similar thing in high school. I did a dual enrollment program where I lived 2 years on a college campus before going to Yale and doing 4 years there. And then really wanted to come back to New York. So I chose this program and now I'm here. And when I'm not doing school things, I mean, my hobbies are like to bike around the city, play soccer, pre-pandemic, going to concerts and stuff. And now more so in the house playing video games more than anything else at this point. So yeah, so maybe we can get started with a poll just to get you guys get a feel of the group. So for me, this is a very crucial question. So if you guys could only eat one food for the rest of your life, would you rather do pizza or tacos? See, this is how I know it's a New York group. It's mostly pizza that's winning. All right. So it seems like a pretty strong consensus, pizza. I do agree with that. You get a pretty balanced meal with pizza. You get your carbs, you get your dairy, you get your vegetables. If you're a meat eater, you can put some meat on there too. But I feel like tacos are kind of cleaner to eat. So if I had to do it for the rest of my life, maybe tacos would feel less heavy to do every day. But I could go either way. All right. Thanks, guys. So yeah, as I'm going through like the lesson, feel free to interrupt me, ask questions. Pizza for lunch, yes, go for it. Definitely feel free to interrupt me, ask questions at any point. And at the end, again, I'll try to leave enough space for Q&A and everything, whether you want to ask about the science or my path in life in general, feel free. All right, so I'm gonna share. So I'm gonna be drawing things in Paint. All right, so as I mentioned today, I'm going to be talking to you guys about cancer metastasis. So, in our bodies, there's kind of a, see if this is going to start drawing or not. Okay, I might have to use my finger. Okay, there we go. Yeah, so in our bodies, there's kind of a constant balance between cell growth and cell death. So here I'm kind of just drawing. If you were to look at your skin under a microscope, you would see kind of this nice balance of cells that kind of grow up from the bottom, we call the basement membrane, which is down here. And then as they grow up in layers, they die off. So, when it's kind of dry out like now and you're kind of ashy, that's all those dead skin cells that are coming off. So there's a balance between these cells at the bottom that have to grow and these cells at the top that have to die. So if you lose this balance, then you could just keep growing these cells and maybe you would have you'd be walking around with six inches of skin, which would not be very good to live with. So this is what's happening in our bodies every day in every tissue. There's a constant balance between cells that are dying and cells that are growing in order to maintain our tissue. So what happens in cancer? I'm just going to make a new one here. So in cancer, instead of having this nice, flat, even layer of cells, you get a cell that kind of goes wild and keeps growing, disrupts all the nice, even architecture we have, and then keeps growing, and that's what we call a tumor. And how does this balance or imbalance happen in that cell? So what happens is that a cell has to take on what we call a mutation. So here's an individual cell, and I'm just going to simplify its DNA in the nucleus here. So what has to happen for a cell to become cancerous and start growing crazy and never dying is it has to have these mutations in its genome and its DNA that turns off a gene that stops cell growth, and that's what we call a tumor suppressor. So a tumor suppressor is a gene that, when it functions normally, stops your cells from growing too crazily. So when you have a mutation in a tumor suppressor, then the cell you're kind of cutting the brakes on that cell, and it's able to keep growing even if the conditions normally wouldn't be right for it to grow. But you have to be unlucky to also get a mutation in what we call an oncogene. So I'm just going to try to write this out. So an oncogene is a gene that makes a cell grow. And in normal conditions, it's not always on because, again, you don't want the cells in your body to be constantly growing. But if you're lucky enough to get a mutation in both a tumor suppressor and an oncogene, then the cell has no limitations to its growth. So you're basically both cutting the brakes and putting a brick on the gas. And the cancer cell just takes off and forms a tumor. So I'm going to ask you guys another question, and you don't have to know the answer by any means. But what do you guys think can be a source of oncogenic or cancerous mutations? So what are things that can cause these mutations in a cell to happen? And choose as many as you think. So maybe I'll give you guys a minute or so.
About 60% voted. We're waiting on about 30% of you all. You won't know if you got it wrong. Just throwing that out.
Just don't feel shy about putting the wrong answer. Okay, so that's a pretty good percentage. So I'll just end. We got 80%. All right, so almost all of you chose radiation. So exactly, that's kind of one of the strongest sources of mutations. So basically radiation, what it does is that it's super high energy and it just hits your DNA and like breaks it pretty much. And so your cells try to put together your DNA to fix it, but sometimes it makes mistakes. And that way you can get mutations that could hit an oncogene or a tumor suppressor. Infections, some of you also voted for that, and that's also correct. So there are certain viruses and certain bacteria that can, when they're left untreated, can lead to cancer by causing, again, mutations. So one example is HPV, which causes cervical cancer. So that's why we have this HPV vaccine now that they call a cancer prevention vaccine because it really does do that by preventing HPV. Another example of a bacteria that could do this is called H. pylori. So it's a bacteria that infects your stomach and causes ulcers. And then over time, as that infection continues, you can develop gastric cancer or stomach cancer. A lot of you guys chose being born with one, so very good. So there are, it is possible to be born with a mutation in one of these genes. So an example that you may have heard of is called BRCA. Or so that was one thing that Angelina Jolie had, and she got what we call a preventative mastectomy. So she surgically removed her breast to prevent the chance of her getting breast cancer because she was born with a mutation in a gene that could increase her chances of getting breast cancer as well as ovarian cancer. So very few of you chose oxygen, and that's kind of a trick question, but actually oxygen can cause cancer. So what happens in our cells is that oxygen can cause these things called free radicals. And our body has a system in order to kind of clear oxygen that kind of changes its structure and can destroy or affect our DNA. So, when you're actively breathing oxygen, like, no, that's not going to instantly give you cancer. But if the process within your cell can no longer clear these free radicals, then it can lead to cancer. And some of you guys chose 5G network. It actually doesn't cause cancer. It's just your normal wireless network. So I know there are probably a lot of conspiracy theories out there about 5G, but 5G does not cause cancer. So don't worry about that. All right. Cool. So, and as you grow older, the chances of getting mutations like this increase. So that's why most people with cancer tend to be older. Just over time, there's more and more chances of you to acquire these kinds of mutations. So, just gonna make a new canvas again. So if you have a tumor that's forming in your skin, for example, that's as long as it stays there in the skin, it's pretty harmless because there's nothing really around. So if you had a tumor in your arm, there's not really anything around there that it can affect much. So the problem is when these tumors start to metastasize. So I'm again going to draw my skin here. And now my cancer is forming. So over the course of years and years, this tumor is developing and growing bigger. And then eventually a few of the cells in the tumor leave the skin and actually enter the bloodstream. And once these cells enter the bloodstream, and kind of look around the body and see, oh, what would be a nice environment for me to go into and stick around in. That's when we have a problem called metastasis. So, for example, say these cancer cells, go around and then they find the lung and they think, hey, it looks like a pretty good place for me to go. So they, once again, exit the bloodstream and travel to the tissue of the lung. And then there, they form yet another tumor. And this is what we actually call the metastasis. So a metastasis is a tumor cancer that has left the primary site. So the primary site being this part of the skin here. It traveled all over the body and found an organ, a new home for itself to grow in. And so this is what kills the majority of cancer patients. So 90% of cancer patients die when their tumor goes to an organ like the lung, the brain, the liver, and grows there and starts affecting the ability of that organ to function. So when you can no longer breathe, where you can no longer, perform tasks with your brain, or your liver can no longer clear toxins because this tumor is growing in it, that's how patients tend to die from cancer. So our lab is really trying to figure out what causes these cells to change their behavior and be able to survive in a completely different environment from where they started. And every step of this pathway, from leaving the primary site to entering the bloodstream, to leaving the bloodstream, to finding a new organ and growing in that organ, there's only a small percentage of cells that can survive each of these steps. So I'll have another question for you guys. And again, you don't have to be right at all. So just take a guess. What percentage of cells do you think What percentage of cells do you think are able to make it from the bloodstream to actually form in this metastasis? So like what percentage of cells make it from here to actually doing this at the end? Again, I'll give it another 15 seconds or so. I'm just seeing the chat now. You guys talking about pineapple on pizza. I didn't do it either. So. Okay, so almost all of you voted, so that's good. All right, so let's go through. So the majority of you guys voted 10%, which is a very, logical guess. It seems like small enough, but also you would think, cells that have already made it this far, they can probably make it towards the end. But the actual answer is 0.01%, which is 1/10,000th. So only 1/10,000th of all the cells that make it to the bloodstream. And again, that's already a very small percentage of the cells that actually leave the main tumor. Only 1 of 10,000 of them are actually capable of going to the lung or whatever organ and growing again there. So, in cancer, it's a constant battle of like evolution and numbers. The numbers of cells that can actually form the metastasis is very, very small, and they have to somehow evolve a number of techniques in order to make it to the end. And so again, that's kind of what our wait, I forgot to share. I don't know if you guys can see the answers. Yeah. So that's again what our lab is kind of trying to figure out, how these cells are able to pick up like little tricks and techniques in order to actually make it to the end of the metastatic cascade, as we call it. Okay, so I'm going to demonstrate to you guys how we actually study this in the lab. And, to really study this process in a way that can be most informative, we have to develop a system that mimics kind of all these steps of both having the primary tumor and then tumor cells that are able to get into the bloodstream and then make it to a different organ. So basically what immunofluorescence is, it's a way of using antibodies in order to amplify a certain thing that you want to look at. So assuming these are all cancer cells this time, these cancer cells have certain proteins on their surface that are unique to them. Again, since they're coming from a place that isn't normal in the lung, so they're coming from the skin instead of the lung, these cells look different than most of the cells in the lung. And we can take advantage of that to actually find the specific cancer cells that are in the lung. So to do this, we use antibodies that can identify these special proteins on the cancer cells. So yeah, these are antibodies. And then we use another antibody called the secondary antibody that has a tag on it. So I'm just going to use a different color here. And this tag is fluorescent, so basically glows. And then we can use a microscope in order to basically shoot a laser at, oops, accidentally stopped sharing. So this microscope can shoot a, let's see, I want to change the color. Yeah. So once this microscope shoots a laser and it turns on this glowing fluorescence, then that fluorescence goes back to the microscope and it's able to show you where the antibodies bound to the cancer cells. So to do that, we basically we cut up the lungs into these slides, and then we add on the antibodies and everything in a process. And after about a day or so, then we're ready to actually look at them under the microscope. So I'm actually in the room now with the microscope. So I'm going to show you guys. So this is our big microscope. So this is actually where you look in. So the actually part where you look in is way smaller compared to like the whole thing, which basically takes up this entire room. And then what you want to save the pictures that we see in the microscope, then we have a computer here that saves the pictures. So like, do you guys think, how much do you guys think that microscope is? So when I heard it, I was shocked. So you can just guess in the, and while you guys are guessing how much it costs, I'll answer these questions about how do we slice them so thin. So there's a machine called the microtome that you pretty much put so it's kind of like those meat slicers. Have you ever seen like a deli meat slicer? It just makes those like really thin slices and the machine just like cuts it. It's kind of similar thing. So you can like program the machine to make it, tell it how thin we want the slices to be, and then put the lung on there, and it cuts the lungs. And in terms of the orientation, it doesn't really matter unless, because usually, whoever's doing it kind of places them in the same way. If you want to look at a certain part of the lung, then it is important to like make sure you're orienting it in the machine so that you can get the part of the lung you want.
Is the lung encased in something harder, or like okay?
Yeah, exactly.
Imagining it sort of swishing under a blade. Yeah.
Yeah. So after I take the lungs out and put them in tubes like this, then we do a process called paraffin embedding. So we basically put them in this kind of I don't know what it's most similar to, like kind of a wax, basically a wax mold, and then from that you can cut through the wax and take some lung with it as well. All right, so you guys have made some good guesses. So the highest was 800,000. Sorry, 100. I just spoiled it. Sorry, guys. The highest was 100,000. It actually costs 800,000 just for that microscope. So it's very expensive to do science, and we spend a lot of money to like figure these things out. And every time I come in here, I'm scared that I'm going to break it, because that would be my whole entire life's earnings, basically. Yeah, so I'm going to show you guys an example of a picture that we get at the end of this process, and then we can do a bit of Q&A at the end. So I have here, so this is a picture I took recently with that microscope. And you see the glowing. So I'm just going to let it play and then actually pause it at kind of the main part. So this is me kind of going up and down through the section in order to get the full dimensions of the tumor in the lung. So I'll just pause it right here. So basically, in order to do this, so as I mentioned, we use these antibodies. In this case, it's glowing green, the cancer cells. We also use a stain called DAPI that just stains every cell in the lung. So it's not specific, but it kind of lets us know that wherever we see green, we know it's real, because there's also a nucleus or a blue dye with that green. Because, for example, like here, it's possible to just have a bunch of green, but it's not actually a cell because there's no nucleus associated with it. So this is kind of how, even though I can't see this tumor by eye, by using a microscope that can go from 10 times stronger than my eye to up to 100 times stronger than my eye, I can actually see in these lungs now the tumors. And I'll do another picture that kind of zooms in more. So this is at 40x, so 40 times stronger than my eye. and now you can see the actual individual cells pretty clearly. So like this one's a pretty good one. Again, this green signifies that it's a cancer cell. And then since there's blue in the middle, I know that it's a real cancer cell and not just, the antibody going wherever. So maybe there's like one more minute for questions, but if you guys have anything else you wanted to ask, I can also put my email in the chat if you wanted to ask about, science or why I chose to do an MD PhD or.
I would love to hear about why you chose the MD PhD.
Yeah, sure.
It's gonna be really hard.
Yeah. So I was actually in high school as well when I kind of learned about the MD PhD process. So like in school, I just always loved science more than any other subject, and biology specifically. And my mom was a nurse, so like she had like these anatomy books that I would look at, and things like that, and I would watch like medical documentaries or whatever. So I was like, okay, I really, really want to be a doctor. But I also really liked the discovery aspect of science. And I liked, watching or like seeing, I don't know, like science documentaries and like hearing scientists talk about how they discovered this thing or like this cellular process or like how this. So I basically like both aspects. I like the idea of like working with patients and like learning about the human body and everything, but also like the idea of being the person who like discovers certain things that nobody knows yet. So I felt really conflicted. So I was like, should I be a doctor or like should I be a scientist? So then I kind of like just like Googling what are different career paths in science, whatever. Then I like stumbled upon MD PhD and like learned that it's actually possible to do both. You can get an MD and like go to med school and become a doctor, but you can also get a PhD and like learn how to be a scientist. And the like programs that integrate that, so that you can pretty much become a physician scientist who like sees your patients and like goes to lab and studies the same things that your patients are being affected by. And I liked being able to both have that immediate impact with like actually working with people and like giving them therapies and like speaking with them like one-on-one, but also like kind of the long-term impact that doing research would have. So like in 10, 20 years, like the research I'm doing might actually have an effect on patients in the long term. So that's kind of why I decided to do an MD PhD. It is a long process. So medical school normally is 4 years. And then you go on to residency and fellowship, which is more training after medical school. And an MD PhD program is 8 years. So pretty much double the time because you have to fit in that PhD. So if you were to do them separately, if you were to do an MD and decide, oh, I actually want to get a PhD, a PhD is on average like 6 years now. So if you were to do them separately, it'd be like 10 years. So like doing an MD PhD program does shorten it, but it's still like a long time. But one benefit is that, again, they're like integrated. So they're really teaching you like how to career-wise, how can you do these both as a career, become a physician scientist. And also they pay for med school and they give you a stipend. So I don't think about it as 8 years of school. I think about it, I'm doing the job because I'm getting paid. So, so that's how I look at it. So yeah.
Any last questions from kids for Noma? I want to make sure to be respectful of your time. I think you said 40 minutes and we're past that mark.
Yeah.
But we will follow up if anyone has burning questions to ask you.
Yeah, again, feel free to email me. I'm happy to, talk. This was like the stage where I really learned about MD PhD, and I think it really helped me to, going to college already knowing about it and helping me make decisions in terms of what to do. So feel free to email me or Ms. Mason, feel free to email me as well and relay things to your students, and I'd be happy to talk more about it. But thank you guys. I hope this was really helpful, and